We are now starting session 5. If you're interested in session 6 on the energy path to net zero, you need to head to the orange track. We are in the green track for the odd number sessions, and we're about to start session 5 titled Decision-Making Frameworks for Early Stage Pharmaceutical Development. This is the session I am most excited about because the co-chairs have such great passion for the track. I watched the co-chairs start being new to SDP and learning how the conference works to becoming experts at assembling a fabulous session. So I wish to introduce to you our two triumphant co-chairs, Dr. Julia Varshefsky. She is the founder of Acompoint. She has over 18 years experience in drug development, spanning from discovery to commercialization and life science management. She is an expert in statistics, advises investment decisions, and coaches startups. And Dr. Meryl Berkner, Vice President of Portfolio Strategy and Analytics at Gilead. She is previously a Vice President at 23andMe and has a PhD in biostatistics from the University of California, Berkeley. Julia and Meryl, the session is yours.
Thank you, Gary. My name is Julia Varshefsky, and I'm delighted to welcome you to session 5, Decision-Making Frameworks for Early Stage Pharma Development. Meryl and I will introduce our session to you, and then our speakers will proceed with their presentation. So let me start by explaining a little bit about the genesis of the session and how it evolved into its present shape and form. As we all know, bringing a pharmaceutical product to market is a risky, lengthy, and costly process. It could take over a decade and a half from discovery to product approval, and it could result in $2.5 billion or so in development costs. The probability of success from phase 1 to approval hovers around 14% in general and around 4% for products in oncology. Mid to late stage clinical development, while still very complex, happens in the context of the chosen indication. And so the questions on the table at that stage and the decisions that need to be made are about the specifics of the regulatory path, evolution of efficacy, endpoints and targets, acceptable safety margins, fine-tuning of the patient population for the indication, and timing of the market entry in the context of the evolving competition. My present close work with early stage companies makes me truly appreciate that decision-making paradigms in preclinical and early clinical development stages are orders of magnitude more complex. First, there's a lot more uncertainty in the technical path to success. The proof of concept is still evolving or is not yet fully established. The product is still getting formulated. Animal to human translation may still need to happen. Secondly, in the face of all that uncertainty, the range of possible indications needs to be mapped out to create a sense of the market opportunity, partnering opportunities, and timelines. And finally, if you're a startup, what makes these decisions particularly hard is that they have to be made with restricted expertise pool and scarce funding. I would like to transition over to my partner in shaping this session, Meryl, and ask her what interested you in the session, and could you share with the audience how it evolved into its present shape and form?
Absolutely. Thank you, Julia. Portfolio analytics, frameworks, and decision-making in early stage pharma has been a main focus and interest of mine, both in my roles in larger pharmaceutical companies, as well as smaller companies. Early stage pharma and biotech development often has challenges of limited information, variability, and uncertainty across all of the value drivers of risk, revenue, cost, and timing. Adding some rigor and consistent frameworks and approaches to these early decisions are going to allow us to further strengthen the decision quality across the life cycle of a program. The decision-making frameworks and goals are somewhat different if you're making portfolio decisions within a corporate pharmaceutical environment versus when you're making them as a life science startup. Our speakers today actually represent both sides across that spectrum. First, we're going to hear from Joe Gatto, who will describe the decision-making that investors are facing when investing in a pharma startup. Then we're going to transition over to Eric Johnson, who's going to speak about the management of uncertainty in the early clinical development from within a corporate pharmaceutical environment. And finally, Brian Hobbs, who works with both corporate and startup clients, will bring home to us how important it is to think about later stage development when designing early stage trials. I look forward to learning the approaches and frameworks from our presenters today. And I'm going to turn it back over to Julia to note any pointers the audience should be aware of as we start today's presentations.
Thank you, Meryl. So in terms of the logistics, we will first proceed with all of the three presentations at once. And if you could hold your questions to the end, we'll have a joint Q&A session. You will be able to ask your questions just like you did in the rest of the conference, just feed them to the chat, and Meryl will moderate the session and present these questions to the speakers. Finally, we will have the speaker session where you will be able to speak live and discuss your perspective and questions with the five of us at the same time. Now, I would like to move to introducing our first distinguished speaker, Joe Gatto. Joe earned his degree in engineering economic systems from Stanford University. He further coined his decision analytics skills by consulting with the Strategic Decisions Group and the Decision Company, which he co-founded. Joe became a national decision analysis celebrity after he launched and became the CEO of StarMine. StarMine developed methodology and analytics that quantified the performance of Wall Street analysts and created stock ranking models. Under Joe's leadership, the company grew to $33 million in annual recurring revenue, and it was sold to Thomson Reuters, which is now known as Refinitiv. Joe is deeply involved in the early life science startup ecosphere, where he invests personally as well as a VC. Recently, he launched a $20 million venture capital firm, Seedfolio, that has invested in portfolio over 50 seed stage companies. A large part of Seedfolio focus is in life sciences. Joe will kick off our session by outlining how he approaches thinking about the uncertainty of the early stage decision-making from an investor vantage point. Joe?
Hi, my name is Joe Gatto, and I'm going to talk about decision analysis for biopharma venture investing. And so here's an outline of what we'll talk about. I'll touch real briefly on how I got here. And we'll look at this from two dimensions. One is the portfolio view, looking from a high level. It's a numbers game, investing at early stage. And to demonstrate that, we'll look at data on returns to seed stage investments or early stage investments. And we'll then get into the implications of that data for portfolio or fund construction. Then we'll dive into the deal view, looking deal by deal, how we go about quantifying deal-specific risk and reward. And we'll talk about the need for very high risk-adjusted return and why that's the case. And our probability-weighted multiple invested capital is the key metric we use to measure risk-adjusted return. We'll talk about tackling probabilities and where probabilities come from, and we'll end with a couple of examples. So very briefly, my background's from Stanford, did a master's of engineering economic systems, focusing on decision analysis under Professor Ron Howard, who many of you may know. Was a decision consultant for 5 years, and then founded a company called StarMine. It was a 10-year project. I was CEO. We grew nicely and had a successful exit. After that, I started angel investing, and now I'm calling Seedfolio Fund 1. That was 50 plus investments in early stage companies with my own checkbook. Recently, a year ago, we launched Seedfolio Fund 2, and that was to leverage outside investment. So it's a $20 million fund, and we'll be making 50 plus new investments. And the average check size that we write into companies is about $300,000. And our primary focus is on both therapeutics and diagnostics. Okay, so let's go to the portfolio view. As I mentioned, it's a numbers game. So let's start with some data. The good news is early stage investing can yield extraordinary returns. For data, we're going to go to a group, one of the groups I'm a member of. It's called Tech Coast Angels. There's 450 members. They're based primarily in Southern California, one of the largest angel groups in the entire country. And if you had invested $25,000 in each of the 182 deals that TCA is invested in, they're now shut down or exited, you would have invested $4.6 million, and you would have had a 4.9x return. Very nice. But 125 out of those 182 deals, or 69%, lost money. They returned between 0 and 1x. 0x meaning you lost everything. 1x means you just got your money back and didn't make a return. 15% of deals, about 30 or so, returned between 1 and 3x, 14 between 3x and 20x your money, and only 2%, actually 1.6 from just 3 deals returned over 100x. So that 4.9x was driven by these 3 home runs. And if you missed those 3, your overall returns in the other 179 averaged only 1.4x. So there's 2 major implications for this distribution of returns to early stage investing, that the few winners that you do have must be large enough to carry the losers. And we'll get into evaluating the winners versus losers in the next section. But for now, in the portfolio section, I want to make the point that early stage investing requires large portfolios. And since we've got decision analysts in the group, let's take a look at this a couple of ways. First of all, let's look at from a Bernoulli's trial point of view. So like flipping a coin or spinning a wheel with a 1.6 chance of success, right? That was our incidence of outliers, as we saw in the last slide. And again, it's really important to have these outliers to carry all the losers. Well, if I did a small portfolio of, well, if I only had one, if I had one deal with a 1.6% chance of being an outlier, then I'd have a 1.6% chance of an outlier. If I had 10 deals, the chance that one or more of those was an outlier would be 15%. And if I had 100 deals, the chance of one or more of those being an outlier would be 80%. So it turns out the 50-50 point is about 45 deals. Now, another way to look at this is using Monte Carlo simulation. So I used the distribution of returns that I showed you before, 69% at 0 to 1x, et cetera, down to 1.6% at over 100x. I use that distribution from which to sample. And then we built portfolios, a 10 investment portfolio, 20 investment portfolio, and so on. And if I built a 10 investment portfolio, then using Monte Carlo, we sampled from this distribution 10,000 times. And it turned out there was a 65% chance of getting a 1x return or better. This line here is plotting the chance of getting 1x, meaning your money back or better. The blue line is 2x, doubling your money or better. And green line is 3x, tripling your money or better. So we focus on the 3x line because as a venture capital fund, I'd like to return at least 3x to investors. With a 10-deal portfolio sampled randomly from this set of deals, we'd only have a 22% chance of returning 3x or better. If we increase that to a 40-company portfolio, we'd be at about 50-50 of doing better than 3x. And if we increase that further to 80, we'd be at about a 70% chance of 3x. So while the expected value is always the same, the median and the distributions shift upward as you increase the number of companies in the portfolio. So Seedfolio is focusing on targeting a portfolio of 50 to 70 companies, where a typical VC may have a portfolio of only 20, 25 companies. Okay, so that's the portfolio level. Now let's take a look at the deal view, quantifying deal-specific risk and reward. And we'll focus on our metric of probability-weighted multiple of invested capital. The formula is fairly straightforward. Probability of success times exit, value if success, times 1 minus future dilution, and divided by the post-money valuation. So let's break that down. POS is probability of success. In simple forms, in this very simple approach, it's probability that you get to a successful exit. The exit given success, and then dilution is future dilution required. So to unpack that, this round I'm investing in, maybe it's called Series C, maybe it's called Series A, there's a certain amount of money, and that'll get the company to a certain number of milestones. After that, they hopefully have demonstrated some success, but then they need to raise more money. Perhaps this will get them through a phase 1, but then they need to raise more money for a phase 2, and then raise more money for a phase 3. If we look at all the future money needed to fund this through to an exit, how much each time they sell more stock, the company will be diluted by that amount. That'll dilute our returns. So that's a critical variable to model, to estimate. And the post is the post-money valuation for this deal. Meaning, and we'll get that into an example. So for us to invest, we have the bar rather high at needing a probability-weighted multiple invested capital of 10x. So in simple terms, a company says that, hey, we'll give you 10x your money if you invest. Well, if they only had a 1 in 10 chance of success, that would be a 1x probability-weighted multiple invested capital. If a company has a 10% chance of success, you'd need more like a 100x outcome, exit given success, in order for it to be a 10x probability-weighted multiple invested capital. So let's take an example. This is a made-up example, but we're assuming a 10% chance of success, a $1 billion exit if successful, future dilution of 37.5%. We're assuming that after this round, they'll raise 10 on 15 and 30 on 50. And a post-money valuation of this round that we're investing in at $10 million, raising a $2 million on an $8 million pre-money valuation. So we apply the formula, 10% times a billion times 1 minus future dilution, or 62.5%, divided by 10 million. Our post-money valuation is 6.25x. So that would not meet our minimum 10x, even though it's a $1 billion exit if successful. So that was a super simple example, and we can get as complicated with this as we want. In reality, what I usually do is somewhere in between what we just saw on that slide and not quite as detailed as what we see on this slide. But one thing we just pay a lot of attention to is the probability of success. We'll find it's useful to break that down into probability that it's found to be safe, efficacious, and separately that the team can execute. And to see if it's safe and efficacious, we want to clearly look at the preclinical data. We tend to invest at the late preclinical stage. And so we could get into different ways of modeling exit value given success. We can get into total addressable market and market share, et cetera. And so depending on the need, we get into that in greater or lesser detail. And just as in our simple model, we still care about the current valuation and the future dilution. Okay, well, where do the probabilities come from? Well, like good Bayesians, we try to be both grounded in the reality of past deal statistics, like this deal is like the other 182, 69% chance of losing money, or other relevant data, only 10% of drugs starting phase 1 get approved. We want to ground ourselves with those data. On the other hand, we also want to be able to apply well-informed deal-specific judgments. This deal is different because, well, whatever reasons may be. But before adjusting, let's look at the base rates. So probability of approval, POA, is the probability of success from phase 1 through approval. And they range by therapeutic area, overall 10 to 12%, 4% for cancer, 12% for immuno-oncology, interestingly. Now, probability of approval is not the same as probability of success. Probability of success is success to me as an investor. Probability of approval is, will the drug get approved, or will the company get one or more drugs approved? So probability of success, if we're going to take this all the way to the end, is probability of approval times probability that they also meet market success times the probability that they meet no other failure. On the other hand, probability of success could be less than probability of approval, meaning they could be gunning for an early exit given a successful phase 2, in which case that actually increases the chance of success. So to be specific, we decompose the probability of approval. Well, so probability of approval is not the same as probability to our success of our investment. However, it's the most important component. So we're going to look at we're going to look at that carefully. So the first is it safe? Is it effective? And can the team execute? And we decomposed the literature to try to understand the relative proportion of drugs starting phase 1 failing from safety versus failing from efficacy. And so we've come up with this rough baseline decomposed tree. Right? If we multiply 60 times 25 times 80, we get to 12% overall chance of approval. Now, why do that? Well, the reason we do that is that decomposition allows us to add our deal-specific judgments. So, for example, this may be a drug that was tried in thousands of humans and was clearly shown to be safe, but it was done for a different indication. So we might know that the probability of safety might be 90% in this case. Well, if we increase that by 50%, this probability of approval is going to go up quite a bit as well. And similarly, we may have something particularly compelling on the efficacy story or both. The team may be particularly seasoned, or they may be green, that we like the technology, but we're not sure about the team. We may lower the chance of execution success. And so I won't read through these, but sort of a bibliography, good sources for published success and failure rates. So as an example, this is an investment we made just a few months ago. It's a novel immune oncology cell therapy, and with a probability of success that we ended up assigning 18% to. And how do we get there? Well, we started from the baseline, 12.4% approval from phase 1 for IO therapies from 2011 to 2020, according to the first of the references I put on the prior slide. Now, we looked at that, and there's reasons that you may want to be more optimistic and reasons you may want to be less optimistic. Factors increasing the probability of success were that, in this case, this is a type of indication where an exit can actually happen on a successful phase 2, which is twice as likely as getting to an approval, since there's only about a 50-50 hit rate in phase 3. On the safety side, 160 patients of other therapies safely received similar cells at higher doses. Their approach initially is going to be intratumoral, which will further reduce toxicity risk. Mice are not humans. However, the mouse data in this case is exceptionally strong. There's always good mouse data, but this is particularly good mouse data. So the large raise on this particular deal was going to remove a lot of funding risk, which is a risk of execution. Right? One part of the risk of execution is, will the company be able to raise money on maybe mediocre results later on? The fact that they're raising $25 million in this round allows us to get well through a solid phase 1 with multiple variants, offering multiple shots on goals. On the other hand, the 12.4% factors decreasing probability of success, that 12.4% base rate really includes multiple indications for immune checkpoint inhibitors like Keytruda. So in that sample, there are multiple different indications for the same. So those would not be really statistically independent. So that might be overstating the probability of approval for IO therapies. So using that 18% probability of success from the prior slide, we continue the story with a $5 billion exit of success. And the type of cancers this is going after, the breadth of cancers that they believe that efficacy will be shown across solid tumors, multiple solid tumors. A very relevant comparable company was acquired post-phase 1 for $5 billion. So if they do what they say that they can do and they demonstrate what they can, there should be a very large exit. The post-money valuation on this deal is 40, 25 million raised on a 15 million pre, and the future dilution, we model at 42%. So running that through our formula, we get a 12.5x probability weighted multiple invested capital, and we invested. As a second and last example, when we lead deals, we can tranche the deal to reduce risk. So here's a very live deal. Literally, we're contemplating this as we speak, and we will be, just last week, we put a term sheet out to the company, and we're going to be discussing it later this week. So to be brief, the company has an artificial intelligence algorithm approach that may be able to predict a particular medical event, going to be particularly vague on purpose, 30 minutes in advance. And the value of being able to predict that for the at-risk population that would wear this device and have the AI calculate would be huge. The market value, the value that the market would place on this is huge. There's not much uncertainty there. There is, however, technical risk. The company has done a proof of concept where they've using lots of sensors in a clean environment has shown great sensitivity and low false positive rate. That's great. However, in the real world, they have to show that they can have their AI perform even approximately as well using far fewer data inputs and in a much noisier real-world environment. So they want to raise $4 million to finalize the algorithm on noisy data and then bring this into the clinic. And so on that $4 million raise, and they have most of that circled, they've got about $3 million of that soft circled from other investors. They wanted us to invest our standard check size of $300K. And then we'd learn that whether it was accurate, and then if they could succeed, if it was accurate on technically, could they succeed from all the other risks that they've got to overcome being a startup? If so, we'd have an 8.4 million return on our 28x return on a 300K. But if they failed, either here or here, we'd lose our $300,000. Now, this didn't quite pencil at 9.4x. However, we decided to go back to the company and say, instead of the deal that you proposed with the $4 million all up front, and I know you got most of it lined up, how about we tranche it? So we put we'll invest 25% up front in tranche one. That's $75K for us, 1/4 of our 300K. And then we can do this experiment to see that basically $1 million to them in total would allow them to do the data gathering and train their algorithm on the messy data and see if they could still be as accurate as they were on the clean data. And if so, then that would release the rest of the $3 million or rest of our investment as well. And then we'd be facing the same outcomes on the line. So we'd invest $75K. If it was shown accurate, we'd invest. If not, we wouldn't. So the difference between these is this bottom branch. Here, there's a 50% chance of losing 300K. Here, we've changed that to a 50% chance of losing 75K only. And so that's going to drastically increase the value of the deal. And we'll find out if they're going to go for that. They seem to be positively inclined because they want to get going. So in summary, we've tried to show that early stage venture investing is a numbers game. 70% or more of deals will be losers, and less than 5% of deals will drive most gains. With that in mind, it requires a large portfolio of about 50 or more investments, and the few winners must more than compensate for the losers. To achieve that, to achieve the fact that the winners will be more than the losers, we're looking for asymmetric upside. We're looking for deals that if they do hit, they're going to be big. And the way we calculate that and operationalize it is using a probability-weighted multiple invested capital and trying to get that over 10x. For probabilities, we start with relevant base rates and adjust cautiously if strongly justified. We decompose probability of success risk to sub-factors like safety, efficacy, et cetera, to better apply judgment. And we tranche deals around de-risking milestones whenever possible to reduce investment risk. That's it. I look forward to taking your questions after the other speakers have presented.
Great. Thank you, Joe, for the great presentation. I would now like to introduce our second speaker, Eric Johnson, the Director of Decision Sciences at Glaxosmithkline. Eric has an extensive background in decision analysis. He studied decision analysis at Stanford University under the mentorship of Ron Howard. After graduate school, Eric spent several years as a decision analysis consultant, serving clients across pharma, oil and gas, and electric utilities. Eric then moved in-house into the pharma industry, initially at BMS, where he focused on helping drug development teams generate options and characterize their prospects to enable corporate decision-making. After BMS, Eric moved to GSK, where he is currently helping to structure a new DA practice and supporting research teams for preclinical or phase 1 decisions. Eric is also the co-author of the Handbook of Decision Analysis and is a fellow of the Society of Decision Professionals. Eric is going to dive deeper into the topic and will describe how he approaches thinking about uncertainty in early clinical phases from a drug developer's point of view. Please take it away, Eric.
Thank you very much. I'd like to share with you a talk entitled Stone Soup, or how to do phase 1 pharmaceutical decision analysis. So the agenda for my remarks here is I'm going to tell you the Stone Soup story, and I'm going to tell you why it's pertinent to DA for phase 1 in pharma. I'm going to show you an example. I'm going to talk with you about one of the hallmarks of it, which is light touch data elicitation and success stories. But before I get into telling you the story, I want to tell you how I even came to this topic. Last year at DAG, I gave a talk about phase 1 pharma analysis with lots of clever analytics. And there was a lively Q&A afterward, but nobody wanted to had questions about my clever analytics. They just all wanted to say, how on earth can you do phase 1 pharma DA at all? When we tried, you can't get the data. You can't even get started. So that's what this talk is about, is how do you get started in a context where it's fairly remote from the outcomes of interest, and it's viewed as being fairly speculative. So with that preamble, I just wanted to sort of share with you that background of why I'm doing this. But first, I want to tell you the story of stone soup. So this is, imagine yourself in 19th century Europe, and your village is, everyone's hungry. It's been a bad year for the crops. And this gentleman in the orange jacket is a stranger and wanders into town, and nobody knows what to make of it. And he puts his cauldron down right there in the middle of town, builds a fire under it, and fills it with water, and puts a big round stone in it. And he's just whistling merrily and doing what he's going to do, and we're all thinking, what's going on? What is this? And he says, I'm making stone soup, and I'll give you all some at the end of the day when it's ready. And we all think, you're crazy, man. You can't make a soup out of a stone. But it's kind of intriguing. So some people sort of lingering around the edges. And after a bit of time, he says, what makes stone soup even better is if there's a little bit of salt and pepper. And so one of the villagers says, okay, I got some salt and pepper. I'll bring you some salt and pepper. And then he's boiling away, and they're still a little mystified at how this is going to work. And he says, what just makes an absolutely perfect stone soup? It got some carrots in it. Okay, well, I got some carrots. I'll give you. Gary says, great. You give me the carrots, and I'll give you a bowl of the stone soup at the end of the day. It's like, wow, this is cool. I get this lovely bowl of, I'm not sure how, but a lovely bowl of stone soup at the end of the day. And all I did was put some carrots. And eventually it goes along, and then, just some chicken. Chicken makes it just sort of a perfect stone soup. Somebody comes up with some chicken, and comes the end of the day, and now everyone's pitched in just a little bit, and nobody feels like they did anything over the top. And Ed's made a nice soup, and he shares a bowl with everybody, and he has a bowl, and it all works out. So that's the story of stone soup. And so I want to talk with you about how that metaphor applies to phase 1 DA. So one of the key hallmarks of it is settle for just the minimal number of input parameters, whatever is easy, whatever makes sense, even if it's low quality. And that's the request for just some pepper and salt. And show the people that you're working with some preliminary insight. And that's getting something for nothing. And that gets them into the game, even when they haven't really put much effort into it. But now we're professionals, and this matters, and you may actually be informing a decision. So Are we really comfortable with this decision based on fairly weak inputs? Well, that's where the tornado diagrams come in, and you all know what a tornado diagram. We're going to test plausible input estimates around the inputs we've got and see if it changes our decision. If it doesn't, then we're good. Then we have a robust conclusion. And in cases where it does, now you can go back to that particular expert and tell them, you know, your bar came at the top of the tornado, makes a big deal, it makes a big difference to what we're doing. And, you know, it would be a lot better if we get that from you, and they feel better about doing the work because they know this is not an idle academic exercise. It actually makes a difference. So that's a kind of philosophic basis for it. And just wanted to share with you an example. This is an anonymized version of a real analysis that we did, and I call the project Mango. And this shows the standard fundamental metrics that we use in pharma. So ENPV is expected net present value of cash flow, which is a kind of a standard value metrics. And in an investment constrained environment, we calculate this PI ratio, which is you divide your ENPV by the amount of investment and call it PI. And so you have to calculate your dev cost in the same units as ENPV, and you get your PI and your incremental PI. And, you know, this is in contrast to what Joe is showing you for venture capital, where he had his PWMOIC, but we're using the standard metrics for pharma. And you put a few bullet points here that you can glean from the analysis to give some color to the analysis, and that gives people a bit of a taste of something for free. And then you do your tornadoes, and that's the key part of making it be valid as well as doable. And so this is a direct tornado for option B, which was the one that was better. I should go back and show you. The better one had an ENPV of 158, which is positive, so that if you believe it, we should go. And option B has 38 million better ENPV value and at a better PI ratio than option A. So again, if you believe it, B is better than A. But now your tornado, you can ask, are those robust? And this is the direct tornado on option B, and it shows you that if any of these uncertainties were at its low or its high end of its plausible range, you're still making money. The only way that you would be losing money is if you crossed over zero. So that's zero line, you go into losing money side of the chart. And we're solidly in the makes money side of the chart. And so, and you point out, as you present it, I haven't vetted all these inputs with all the experts, so some of the uncertainty bands here are very wide. But even so, they don't cross zero. That 158, I'm not going to tell you it's the right number, but it seems pretty likely that the answer is north of zero. And then you add some more insightful bullet points here to give people a sense that, you know, what you're doing is a value. In this case, I showed what were the range of assumptions on the key parameters, and they're pretty wide. Then the next thing you do is a difference tornado. And you remember that there was a $38 million difference where B was 38 million better than A. So this tornado is a difference tornado, looking at that difference, and it shows you how much would that difference be if the various variables did what they're going to do, varied according to their nature. And so now this zero line is a crossover point between favoring option B, which we're out here with a positive difference, favors option B. And if you cross to zero, it means that under that case that crossed zero, that would have been an argument for option A. And in this case, and in a surprisingly large number of cases, I'm finding, it doesn't cross. So not only is it a robust finding that you should go rather than no go, it seems to be a robust finding that B is better than A. And you can put some commentary here about what's going on in the particular variables that make a difference here to give a little bit more insight to the people that you're working with. And they may find this puzzling because they saw $100 million worth of swing attributable to product profile. They say, why doesn't it cross zero here? You only got 38 million to play with. And a key insight is that many of these variables affect the two options in roughly equal amounts, so it cancels out, and that's why the finding is robust. So just to recapitulate, you got two main decisions in early phase: go, no go, and then choose how to go. And your direct tornado tells you the go, no go is robust, that you should go, and the difference tornado tells you that the choice of how to go is also, in this case, robust. Now, how do you get those initial estimates? This is the ask for just a little bit of salt and pepper. And this is kind of tailored to GSK, but I think is relatively constant across the industry. You start with a middle estimate, and then you get your high and low range. And I know that Kahneman and Tversky will say, well, yeah, you risk having inadequate adjustment from your anchor. I'll take that challenge. So you get your middle number, and the cost and the timeline, the project manager normally has to do that, and they have to put it in the system because everyone worships the cost and the timeline. Then the four pillars are cost, timeline, probability of success, which we call PTRS in the industry for probability of technical and regulatory success, and peak your sales. There are other parameters, but those are the four main ones. So you've got cost and timeline for your project manager. We in decision science are empowered and required to make a PTRS to elicit that from the team. So that's going to be there. And then for your peak year sales, that can sometimes be tough, but often you can find some kind of an estimate. There may be old numbers floating around that aren't perfect, but they're better than nothing. And then for the ranges, this is the part where I think is important, is get some ranges. If the team leader is friendly toward DA, get it from them. If not, make it up yourself. Now, why am I telling you that you can go make up these numbers yourself? The reason is, let's look at it in the context of the great scheme of expertise. If you have the right expert, their range on that parameter can be very tiny because they really know what they're doing and they've done a lot of homework. If you ask the man in the street, they got to give you 4 orders of magnitude because they have no clue what that parameter even means. Now, where do you as a decision analyst stand? Well, maybe you're here. You're smarter than the man in the street. You've been around the business. You've seen other analogous projects. Where does the team leader stand? They're a little closer. They're not as good as the expert, but they're a little closer. So all of us are experts. Some are better experts than others. Now, bear that in mind as you work with your whoever is the available expert on this, and you use your judgment as a decision analyst on how wide is that range, and you make it wide enough to reflect the level of expertise that they have. Make it good and wide. If that still doesn't cross, if those tornadoes still don't cross zero, then go forward. If it does, then that's the big blinking red light that says, wait a minute, let's really look harder at this one. And a key aspect of that is make so few inputs that you can put them all on a slide in 18-point font, as I've done here. Just absolutely the fewest number of inputs that you could get by with. So you can put every last one of them in the tornado diagram, as I did in this case. I mean, you didn't see this many bars in it, but they were all there. It's just that I didn't show you all the little tiny bars at the bottom that have even less impact. Very few parameters. Now, this is sometimes contrary to the culture in a pharma, because by the time it ends up in a profit and loss statement, it goes year by year by year from now till 2040 or 2050. And everyone thinks if you just make a peak year sales and a time to peak and you fake in the year by year data, how can that possibly be right? Well, the answer is, if you are responsible about your parameters, the range of curve shapes that you get will include the truth. And it's far better to parameterize these inputs so that they can all fit in the tornado and you can find out if they matter, than it is to put in false precision and go year by year by year data. Because if you're getting year by year by year data this early in the project, it's probably wrong. And it's better that you would be wrong in a way that you can see the impact. And don't be afraid to invent a fudge factor, a multiplier that addresses something else for which you don't have specific expertise, like the impact of some competitor launch on your peak year sales, or what happens if you had price interference and you had to take a price hit. Make up a factor, put in a range. If it's wide, go talk to somebody about it. If it's narrow, don't worry about it and go forward. And I think one of the things, if you told me this story a couple of years ago when I was working in late phase, I'd be thinking, yeah, but most of the time, those tornado bars are going to cross zero. And indeed, they do sometimes. And if they do, you got to either call it a toss-up and say, sorry, guys, I can't give you any insight on this one. It's too close to call. Or you have to sharpen your pencil and see whether a little bit better expertise would make it determinate. But I did a convenience sample of the last half dozen projects that I've done, and 5 of them gave a clear answer, and there was one that was a toss-up. Now, I don't really believe that. I think if you take a larger sample, you're going to find that something closer to 50% of them won't work, but 50% or X number of them will, and it's worth doing. And if you haven't spent much of people's time, then it's worth doing to see if you can get that insight. So just to sort of wrap up and reiterate, if you're trying to support early phase pharma, economics is a great way to do it. You can do probabilistic economics, DA style, using the stone soup approach, where you ask for just the minimal amount of inputs required, you put it in a tornado, and you find out what matters, and you focus on what matters. Thank you very much.
Thank you for a great presentation, Eric. Now I would like to introduce our final speaker, Professor Brian Hobbs. Professor Hobbs is presently a tenured faculty in statistics at the University of Texas Dell Medical School. Prior to this appointment, he held positions with Cleveland Clinic, where he founded a section on cancer biostatistics, and the University of Texas MD Anderson. He obtained his doctorate in statistics from the University of Minnesota. Professor Hobbs is a nationally recognized expert in the field of oncology research and applications. He has led the National Cancer Institute's Clinical Trial Design Task Force to arrive at consensus recommendations for first-in-human cancer drug trials for seamless designs. He has been invited to speak and publish and received numerous awards for his work in the area of master protocol designs and tumor agnostic trials. In 2018, He co-founded Telperion, a precision analytics company that works with pharma and biotech sponsors, bringing nuanced insights into clinical program design, execution, and inference by combining data evidence from across a multitude of data sources with machine learning. In his talk, Brian will tie together the implications of the early phase design to later stage clinical outcomes and probability of success. This will bring home the point of how important it is, even when you are in the early stages of clinical development, to think ahead and map out the full clinical development plan that will lead to approval and support commercialization. To you, Brian.
Hello, my name is Brian Hobbs. I'm an associate professor at the University of Texas. I'm an expert in designing cancer trials and Bayesian methods. My presentation is called Moving Beyond 3+3, Design Innovations for Precision Oncology and Implications for Decision Makers. This presentation might be a little different than the other ones in this session. I'm focused on biostatistics and how we can design trials for precision oncology that will improve decision-making. So we all learned about the discrete phase clinical research paradigm in oncology, where we select an MTD from a very small phase 1 trial, typically enrolling 20 to 30 patients. After that, we run a phase 2 trial to evaluate the preliminary efficacy of the drug that we're developing. If that looks promising, we expand to a randomized controlled trial in phase 3. This paradigm has changed in recent years with the advent of precision medicine. Regulatory policy has changed in the United States such that we don't require actually a randomized trial to get conditional approvals. We also have the acceleration of cancer biology yielding promising targets that have yielded exceptional responses, leading to conditional approvals after phase 2 and even accelerated approvals after large phase 1 trials. Additionally, with the innovations in molecular biology, we now have many targets that exist across multiple tumor types. Typically, these targets were developed individually for individual tumors. Many of them are quite rare, however. Now we have the issue of trying to understand whether the targets are effective across multiple tumor types. And we refer to those drugs for which they are as histology agnostic drugs. So with these innovations in regulatory policy and in drug development, we have a lot of innovations in clinical trial design. Master protocols have been proposed as a way to sort of adapt the clinical research paradigm to precision medicine. We have different types of trials that are referred to as master protocols. Umbrella trials, for example, look at multiple different targets. A single target across multiple drugs. Basket trials, however, look at a single target across multiple tumor histologies. So with these innovations, this discrete phase paradigm has changed, especially the phase 1-2 portion. We see clinical development programs in oncology now for precision medicine, they now look more like this. We still have dose escalation in what's known as phase 1A. However, it might be difficult to select a dose, especially for non-cytotoxic drugs. We may not have the specific monotonic dose toxicity and efficacy relationships that actually are the foundation for the 3+3 design. For non-cytotoxics, we may have to take multiple dose levels into expansion cohorts. Now, before the advent of immunotherapies, about half of trials had expansion cohorts, and they averaged about 17 patients per expansion cohort. We now see a massive expansion of dose expansion cohorts in this setting. We might take multiple dose levels, and we may evaluate the expansion cohorts in multiple tumor types. Again, this is in the context where we have a drug and a target that exists across multiple tumors, and we need to understand which tumor types are most promising. We may also need to continue to evaluate the dose and scheduling. So these expansion cohorts are often not rigorously designed, and I'll discuss that later. If dose expansion looks promising, if we have promising efficacy signals, we may go seamlessly into phase 2, and this may be a basket trial, or we're actually developing a trial that enrolls multiple histologies and evaluates the effectiveness individually within those histologies. phase 2 may be tissue agnostic, where only the target is required for eligibility. So we don't actually pre-specify tumor types. And some companies are actually skipping phase 2 and going straight to phase 3. So this is how the paradigm has adapted to the precision medicine context in oncology. And just to give some examples, the Keynote trial had 8 expansion cohorts, and they enrolled over 1,000 patients in those expansion cohorts. And this was the first in human trial. Right, so very different from what we saw before with cytotoxic drug development. We see atezolizumab having 9 expansion cohorts. We see the breast cancer cohort and a bladder cancer cohort with 100 patients, as well as non-small cell lung cancer. We see multiple dose levels being continued to be evaluated in these expansion cohorts. So these are examples of how the early phase has changed. We have to keep in mind sort of the end goal, which is phase 3. Ultimately, we want to demonstrate that these targeted therapies are significantly better than standard of care. But in oncology, phase 3 trials do not have a good track record. You can see lots of papers like this one, why do phase 3 clinical trials fail so often in oncology? Again, it all reviewed 235 published randomized controlled trials. This was mostly in the cytotoxic and early targeted therapy trials, and found that regulatory approval was low, roughly 35%, much lower than other areas of medicine. And they discussed the reasons for this. Now, we've been told that with precision oncology, with tumor agnostic drugs, that this should improve, but we haven't seen that. There were 2 high-profile withdrawals from accelerated approval last year, one being atezolizumab's accelerated approval for bladder cancer, another one durvalumab's withdrawal from accelerated approval, again for bladder cancer. So we don't understand everything about the targets and the histologies, and there's more work to be done in terms of how we design these early phase trials so we can make better decisions. So that's where I'm going to talk about some potential solutions and what's happening sort of in the statistical design world about how we can improve design for precision oncology. So this is the paper that's in press with JCO Precision Oncology. We've thought about how do we optimally design these expansion cohorts, and specifically we're using Bayesian predictive probability monitoring of the data as it acquires. So, it's been reported that, roughly 74% of trial protocols now have an objective for a dose expansion cohort, but they're not planned in advance typically, and they're not designed with statistical rigor. We need guidance on how to design these design expansion cohorts. And we believe that this solution will lead to much better decision-making and more efficient design. What I'm going to discuss in the next few slides is actually available as open source software with the PPC package. Okay, so going back to the atezolizumab case study, if we look at their phase 1 trial for bladder cancer, they enrolled 95 patients with bladder cancer and treated as monotherapy in their expansion cohort. Their protocol says that they were comparing to a null hypothesis of 10% response rate with an alternative of 20%. With these 95 patients, we see that they actually have a type 1 error rate of 0.005, and their statistical power is 53%. So we can see that this is an expansion cohort that's designed for assessing objective response rate. They gave us targets, but the calibration of the design it's not appropriately calibrated for decision making. Under the null hypothesis, now we've done simulation studies to evaluate this design. Under the null hypothesis, on average, they would treat 76 patients. So the drug does not exceed their target. On average, they would treat 76 patients under that scenario. Under the null alternative hypothesis, they would average 92 with this design. So if we look at these 2 designs here, one we call optimal accuracy, the other one we call optimal efficiency. These are the designs that we are proposing that should be used to monitor these expansion cohorts. You can see that we can calibrate these things so they have a better trade-off for decision-making for objective response. We have a type 1 error in the efficiency design that's 4% and a power that's 72%. Now, this is important. The average number of patients we would actually treat when the drug is not effective, according to the target, is actually 32 versus 76. The average number we would treat on the alternative is 78. So you see that spread is much bigger. And that's really what we should be optimizing over. The optimal accuracy design has higher power and also higher type 1 error rate. We would also treat more patients under the null hypothesis, but more under the alternative as well. So just to give you a visual representation of this, what I'm showing you here is like an ROC curve, but we're optimizing over average number of patients treated when the alternative is true, so the drug is effective, versus the average number is treated when the null is true, so the drug is not effective. You can see that these are potential designs that somebody could choose in the gray dots here when the sample size is 95, as shown with what they actually treated in that cohort. The red dot here is actually the optimal design. It's the closest to this point here where we're trading off, treating a lot of patients when the drug is effective, treating as few patients as possible when the drug's not effective. So this is the optimal design. The triangle gives the protocol design. So this is actually the design that was used. So you can see that this is far from optimal. And so this is one way that we can improve decision making. We need to evaluate lots of tumor types potentially to understand their effectiveness. But what we can do is we can evaluate multiple tumor types and have efficient designs to understand which are effective. Another design innovation that has been proposed is randomization across dose and schedule in these expansion cohorts. You can see this paper by Iasonos and O'Quigley discussing this topic in the British Journal of Cancer, just published last year. So they discuss two different possibilities for introducing randomization to better understand schedule and dose. So one possibility is we do dose escalation as usual. For example, there's 5 dose levels here, so we're stepping up. There's too many toxicities here. We go back down to 3. And then at that point, we choose multiple dose levels among those that are safe, and we randomize to refine the dose and schedule. Again, this is important in the context of non-cytotoxic drugs where there's not a clear monotonic relationship with dose. Another design they discuss, which is even more efficient, there's no clear separation between dose exploration and expansion. In fact, we backfill patients after a dose has been established to be safe. We actually backfill patients with randomization to the safe dose. And so randomization is done along the way, conjointly with the escalation. The MTD is not actually selected until the end of the study. So the randomization is used to actually select the MTD. So they discuss sort of the standard thing is to select the MTD and then do no dose expansion. They discuss the objective here. Of course, if we add dose expansion to dose escalation, we get to identify which histologies might be promising for efficacy and identify maybe the appropriate population for the phase 2 trial. But if you integrate randomization, then we can also look at potentially differences in dose and schedule across those subgroups. And again, this randomization is not designed for hypothesis testing. We're not looking for statistically significantly better treatments, but we can get signals about, dose and schedule and population. Finally, the last sort of innovation that we have in this context, thinking about precision medicine and design, is actually understanding heterogeneity across tumor types. So we published this paper in Nature Reviews Clinical Oncology last year with my colleague David Hong at MD Anderson. So we have methodology that will actually dissect and characterize how heterogeneous a targeted drug is across distinct tumor types that have those targets. So I'll discuss, and so this is relevant to the basket trial design context where we have a target that exists across multiple indications. We want to study all of those indications. We also want to understand, does that drug work in all indications that have the target? You can see that this is a table from a paper in JCO that I've written recently. There's many basket trials that have been implemented and that are running now. You can see here that, we have anywhere from 10 to 40 histologies that are being enrolled in these trials. Moreover, there's an average of about 8 patients per given histology for these trials. So it's very complicated to understand. You may have a small number of patients in a particular histology. You may have to make a decision on the basis of that, and understanding heterogeneity is central to that. So we have developed methodology that defines which patients can be averaged from these histologies. We published designs for this. We also have open source statistical software that characterizes the heterogeneity using Bayesian modeling. So just to give you an example, if you start out and you have no data, you may make the assumption that your histologies are what we call exchangeable, meaning that they can be averaged. Now, our models are graphical models, so that's represented by this graph. The patient, all tumor types are kind of equal distant from each other. But as data starts to accrue, you can see that we start to sort out the heterogeneity that we see across the tumor types. Now, this was an example based on the venirafenib basket trial for BRAF mutations. You can see that our method was able to identify that histiocytosis, thyroid cancer, and non-small cell lung are promising, and they're acting together, whereas the colorectal baskets are not. They're not effective. So this is not a tumor agnostic drug. We see heterogeneity across the tumor types. This is another example of the SUMMIT trial, where really only the breast population was effective. Okay, so these are some papers that describe this methodology as well as the software that we've developed. Okay, my final slide. So oncologists are confronted by a complicated landscape. Designing trials for precision medicine is challenging. We can gain efficiency through Bayesian predictive probability monitoring of dose expansion cohorts. We can use randomization to improve selection of dose and scheduling. And we can use the basket trial framework along with appropriate methods to identify tumor type heterogeneity. Thank you.
Great. Thank you for the fantastic presentations today. So now I'd like to welcome all of the speakers and Julia to our Q&A session. We're going to spend approximately the next 15 minutes on Q&A and welcome everybody to send questions via the chat function over to us. And Julia and I will be monitoring that and asking these questions to our various speakers. So I'll go ahead and start with a question here. This is for you, Joe, and it's about the team factor. You spoke a bit about a variety of different factors that go into your modeling, and one that's extremely important is the team, the founders, the management team, the board. And would love to get your perspective on how you think about that dimension. And if you have any formal modeling about that, that actually goes into your assessment, as we appreciate there is a lot of variability as well as it's maybe hard to quantify.
Sure. So I agree that team is, of course, super important. There's a little bit of a binary decision and then there's a sliding scale. The binary decision is if this technology were my company, would I hire this person, the CEO, and or this team to run it? And if not, if I'm not excited about that, it's probably just going to be a no-go. So no numbers are going to really get over that. And, so that's a judgment. It's subjective. I've got a little practice as a CEO. I interviewed 1,000 people and said no to 4 out of 5 and hired 200 and stood over 10 years the consequences of those human judgments. So there's a little experience there. But then within that, if they could be, good founder market fit and a lot of things in the right direction, and where I would give them a shot, then it's a matter of, as we talked about when we decompose like the probability of approval to safety, efficacy, and execution. Execution is a big factor. And so where that might be 80% for someone who's done it once before and 90% with multiple successes, if this is someone's first rodeo, it may be down to 60 or 70%. So those are some ways in which we take it into account. And on the binary, it could also be the board, not just the CEO. It could be a dysfunctional board and one that the CEO is unwilling to change. And those could be showstoppers as well.
Wonderful. Thank you, Joe. So now I will look at a few questions here from the chat. And one of them, Brian, since you're the last presenter here, wanted to ask one directly to you about how do innovations in trial design in the earlier stage affect stages of drug development in precision oncology, if at all? So I'd love your perspective on that.
Sure, absolutely. Thank you very much for the question. So I would point you to just that there's many case studies where you can ways you could answer this. I would point you to the Amgen drug, sotorasib, that was just approved for KRAS. If you look in the New England Journal, there's an article from October written by FDA authors where they assert that they did not do robust dose evaluation in the early phase, and they're actually requiring a post-market dose exploration for that drug. And with that article, the FDA is advocating for more and more dose exploration in early phase for oncology. So that's just the dosing part. If we think about, tumor type selection, so I've helped design a lot of tumor agnostic trials at this point, and you may not have all the data that you need going in when you design the trial, but you want to give yourselves opportunities to accelerate development for particular indications that that have marketability, right? So typically when we work with investigators, we think about there's preclinical evidence and the target is very prevalent in these particular indications. But if we were able to hit this indication, that's a big market. So I've been helping design basket trials where we have multiple indications and we're monitoring the data. And then we have decision rules at a certain point to accelerate particular indications. So all of that, selecting an appropriate dose, making sure you've explored the dose space effectively in early phase, as well as getting to the right indications that you want to accelerate forward in later phase. I mean, it's critical to later phase development. I would add on top of that, if you look at the Tizo and the durvalumab examples in second-line bladder cancer, both of those trials got accelerated approval on the basis of uncontrolled phase 2 trials. They showed proof of principle through the mechanism. So they had targeted therapies. The targets did better than patients that didn't have the target, but they didn't evaluate if that target was how that target would perform under the chemotherapies that they used as standard of care. When they got to phase 3, it turns out their biomarker targets, that population did very well with chemotherapy. So they didn't have successful phase 3s. So there's this push now, and can we bring in external evidence in some way? Can we bring in real world data about survival, not about tumor response, but about survival? And can we use that to kind of understand if the targets we're developing are also heterogeneous for the standard of care that we have to go up against in phase 3? So all of these decisions tie into have implications for later phase.
That's very helpful. Thank you very much. And actually, just as a follow-on to that, your focus, of course, is in oncology. Have you seen any of this being used, these sorts of concepts outside of oncology? There was an audience question on kind of understanding that other therapeutic areas as well.
For sure. And it is a different space, right? So one of the things that is pointed out in the New England Journal paper, I think, Dr. Pazder is the senior author of that paper, is that randomization to dose is common actually outside of oncology. It's just uncommon in oncology. I think that has to do with we are reclassifying cancer. We are subsetting cancer into smaller and smaller subsets of patients. That means it's harder to run trials. That means we really need to, we're running trials with potentially heterogeneous subpopulations. It may not be true that because a patient has a target, that the drug is effective in all indications. And certainly it's more rare to see the tumor agnostic drugs. Mm-hmm. So I think that so oncology has some unique aspects, and I think that that's why I just read a meta-analysis of master protocols where 86% of the master protocols that have been implemented have been done in oncology settings. So we really do have to sort of, we're kind of in the small evidence world, right? We're in the limited evidence world in oncology, but that doesn't mean that we shouldn't have appropriate design that facilitates better decision-making.
That's very helpful. Thank you again for your perspective. And I'm actually going to jump to one of the most recent questions here for both Eric and Joe. And I'm a little biased here because PTRS and probably success are in my daily life and work at Gilead. But the question here for both of you, perspective on this is, PTRS, so probably a technical and regulatory success, or POS, still relies heavily on human judgment to evaluate factors that make the asset more or less likely to succeed than history or than generics. What are your thoughts on the use of AI or machine learning techniques to derive these sorts of probabilities? Let Joe and Eric comment on that.
If you have anything.
Well, so just briefly, I've seen some, AI approaches, and they can be into two categories. One are black box answers, and it's a little bit hard to reason back to how they got there. And others are more clear box, where you can see the factors that got weighed along the way. So it can be AI can be a great assistant when, especially when it can, expose pockets of the data that may not have been obvious to someone might be using high-level summary statistics like I might be using. So it can be a great tool, especially if it can amplify the human judgment, not replace it.
Yeah, it's a fascinating thing to look at, and I encourage people to have a look at it, and I am interested in finding AI that is explainable so that it can be part of a conversation where we're engaged with human activities.
Can I make a comment on this?
Yes, please do, Brian.
And maybe not specific to probability of success, but just in the context of clinical research or biomedical research. We have to remember the AI techniques that came out of Silicon Valley were made for marketing purposes, right? We have millions of web traffic data, and they're trying to predict if you should click on this ad, right? Or if you would enjoy this website or something, right? So the risk of that is very different than drug development. And they're averaging over millions of data points. When we take these algorithms and develop them in clinical research, interpretability is the foremost importance. They don't need interpretability when they're trying to give you an ad or push an ad your way online. So a lot of the development in the space that I've done is trying to understand patterns in the data that we can take back to the clinicians and the scientists and say, these are the patients that are responding. This is the subpopulation, and we understand that subpopulation. And it's very different than saying, than the predictions that are done on the web, right? So.
No, that's a very, very valuable point there. Great. Thank you all for your contributions on that answer. I'm going to actually go over to you, Eric, on decision analysis and pharma, and how, there have been challenges sometimes for decision analysis to get a seat at the table, to be able to get a seat at the table, and especially possibly in early stage pharma. And so would love your perspective on your experiences on being able to prove the value of this and get a seat with those decision makers at the table. Oh.
The seat at the table, it kind of depends on whether there's a demand for it coming from the top down. I mean, that's what the table means, is who controls the table. My team leaders like it when I come around because I can explain to them that you can make a smarter call, you can test your intuitions about which way to develop your drug, you can make a more compelling case, and that works because the executive vice president who makes the call will pay attention to the economics. If they don't, forget about it, go to work for a different company. So in terms of the seat at the table, it's whether the decision maker wants to hear it, wants to decide that way.
No, that's very helpful. And actually a follow-on question to this is separating out from the seat at the table, but actually one in this field works with a lot of collaborators across the organization for the various value drivers. So as you outline them, whether it's the timeline, the cost, the peak sales, in the risk, of course, that may come directly from your group, these inputs come from a variety of different groups around the organization. So there was an audience question on how does your approach go over with your colleagues? And I think what the question is asking is, especially in terms of roles and responsibilities, where the market analysts and the finance collaborators may feel it's their role to be providing some of these insights. And would love to know your experiences on how you've collaborated cross-functionally.
Yeah, and I mean, I saw the question that came from Jay, and I think Jay's been here before, and he knows that it's commercial, is where you get the pushback. Commercial has a cult of secrecy, and they won't share the numbers, and it makes a huge difference, and it's the biggest problem. And so there's no easy answer. I mean, try to find numbers that have some pedigree, some previous look at this compound, and reuse that as your base case and put a wide range around it and caveat it carefully and say, yeah, and sometimes you need that footnote there. These numbers are not governed by the commercial line, and these are unofficial. They're directional to one's point of view only, because it's going to be that kind of pushback.
Great, no, thank you for that. I'm going to pivot over to you, Joe. And this was a question from the audience on how you determined that 10x was the appropriate hurdle for inclusion in your portfolio, and would 8x, some other x, be inferior to that, and how 10 was derived?
Yeah, good question. So I can answer that a few different ways. On the one hand, it feels about right. That's a really squishy qualitative answer. But here, what do I mean by that? If it was higher, if it was much higher, I wouldn't be, not too many deals would be getting through, and I wouldn't be doing any investments. And if it was much lower, I'd be making too many investments. So, it just in terms of, in the context of my deal flow, and which is very high, it seems to let about the right amount through. Another is kind of the follow-up question that one should ask, especially if they were thinking of investing in my fund, would be, does that mean you're promising that you're going to return 10x to investors? No, of course not. I'm aiming for 3 to 5x. And so why the disconnect, right? Where's that all going? And I realize that I'm human, and I've got biases, and those biases are going to be asymmetric, right? The fact that I do what I do is there's optimism behind some of my judgments, and so it's a margin of safety, if you will, that if I end up returning 3 to 5x to investors, that'd be great. But I got to keep the bar high to counteract my optimism. And the third reason why it ends up being useful is it's often been the case, at least in 2 or 3 different cases, where I'm always open book. I'll show the entrepreneur, here's the probability. And at first I'll ask them, if I'm using this probability, would you agree, higher or lower? No, well, we think that's fine. And exit given success. And so I vet all the numbers with them. I said, well, here's the answer. And it only comes out to an 8x, right? Given the price you want, a $10 million valuation. We can't get there, but I need 10x. But if you could lower the valuation to 8 million, then that would work. And then they say, well, I'll have to talk to the board. And then they come back and say, well, okay, we're willing to lower the valuation to 8 million. So those are 3 reasons why 10x has worked out pretty well for me.
Very helpful.
But follow on that, how do you come up with that future valuation? Because that in itself is could be a very huge range when you're trying to do it ahead of time.
By future valuation, you mean the exit value if given success?
Yeah, the exit value. I mean, the range there could be in the billions that early on, right?
Yeah, you're right. I mean, that's, I could probably talk about that for 15, 20 minutes. We don't have that time. So look, is this a first in class, immunotherapy that if it works, it's going to, be addressable against 30 different types of solid cancers? We're not talking about a $100 million exit if that's the case, right? We're talking about multiple billions of dollars. Is this an orphan indication with only 5 years left on the patent? Well, that's not going to be $1 billion, exit. So, you take into account the market size. I sometimes do the MPV models from pharma point of view to take into account future development costs. If this is an early sale, peak sales, et cetera. So, it's art more than science, but it's using relevant comps.
Okay, got you. Thanks.
Great. So I'm being mindful of time here. I appreciate we are at the end of our Q&A session. So Gary, I'm going to turn it over to you. I know you have a short announcement before we move into the speaker breakout.
Thank you, Meryl, and thank both you and Julia for creating this excellent session. It's been a pleasure to work with you over the past few months. So 3 things to mention. Immediately, you can go to the Meet the Speaker breakout room for this session. Meet the Speaker lasts 15 minutes, but if you and the speakers want to stay longer, you can stay as long as you wish. Second, after Meet the Speaker rooms, we have a 30-minute conference break. Then we're meeting again at 1:30 p.m. on the West Coast or West Coast time and 4:30 p.m. East Coast time. And third, during the break, Dave Matheson of SmartOrg will be in the sponsor room discussing how to apply DA concepts to innovation. So yesterday, Dave presented a fabulous talk. And if you want to learn more about supporting innovation with DA, please visit Dave in the sponsor room. So in sum, meet the speakers for this session, go to it, it starts immediately. Then we have Dave in the sponsor room, and however you spend your time, please come back at 1:30 West Coast time, 4:30 East Coast time for our next and final session of the day. Thank you.